So I want to welcome everyone this morning. Thank you for coming and participating in this presentation. It's a presentation specifically designed for, hopefully for librarians to learn enough information about this project that you can help researchers in your institutions. So it's a bit of train your trainer kind of presentation. There are two people on the call that I wanted to bring your attention to. One is Jeff Grete, who's going to be presenting today, and he's the PI of the project along with Marianne. And Kawei Lin is the project manager, and she is also on the call and will help with questions at the end. And I'll put both of our emails in the chat if you have questions after the call. But welcome and thank you so much for joining us. On to you, Jeff.
Okay, thank you, Stephanie. As Stephanie mentioned, I mean, this is really trying to do outreach to, as she said, train the trainers, and not just for researchers. As you'll see as we go through this, a lot of this can also be applied to students, postdocs, trainees as well. So, the resource does have a pretty broad reach in what it can provide. And so, start with a little bit of an overview on dkNET. It is funded by NIH, by the National Institute of Diabetes and Digestive and Kidney Diseases. And it really is meant to be a research resource portal for, biomedical researchers, for clinical researchers as well. And as I mentioned, students and the like. And really, it's meant to be a way to connect these researchers and NIH funded centers to resources that they may not be aware of. And it is maintained by our laboratory at the University of California, San Diego, and that is run by Marianne Martone and myself. So who is dkNET'S target audience? Since we're funded by NIDDK, our primary audience is basic and clinical researchers in diabetes, digestive, obesity, endocrine, metabolic, kidney, urologic, nutrition, bone, and blood diseases, really the whole gamut of what NIDDK covers. However, as I already sort of alluded to, the tools and services are relevant across all biomedical domains. And as you'll see when I go through some of the examples later, when we discuss some of the reports, if an antibody is used in something that's specific for NIDDK, it could also be used for something that's specific for, cancer research, for neuroscience, and the like. There are 5 primary areas in which we are providing information right now, and we will go through each of these areas. But the most important aspect of this is that on the next slide, these different items sort of apply in various aspects of the research process. So as you notice, as one sort of goes and plans an experiment, constructs a hypothesis, collects and analyzes data and publishes the results, what we've outlined here are where these various areas can tie in. So for example, when you're doing some background research, our discovery portal, which brings together search across 200 plus different biomedical databases, allows a researcher to go in, and search for information, for data, for funded grants, funding opportunities, literature, tutorials, things that might be needed when one actually, is doing some background research. There is the Hypothesis Center, which is one of the new areas that we're building out on the site, which can allow people to interrogate and examine data to help construct hypotheses. When one is actually then planning an experiment based on hypotheses, our resource reports, which provide information on specific resources such as antibodies, tools, and organisms, can be used to help a researcher make a more informed decision, about what resources to use. And then for NIH's Rigor and Reproducibility mandates for grant proposals, we can also then use that same information to help provide authentication reports, which provide assistance to researchers, in terms of, what validation methods they should provide. When you're collecting and analyzing data, again, the resource reports might come in useful in terms of finding additional software tools, related to some tools that you're already using in the lab, as an example. And then when you publish your results, you need to know, with the new NIH data sharing mandates, that are becoming more rigorous, as we move forward, where should, I publish my data? Where can I share my data to meet these NIH and NSF guidelines, that are coming out. So the portal itself, when you come to the landing page, these various areas are sort of right there on this front page, grouped into the various categories that I went through just now. And when we sort of start our tour, we'll start with the resource reports, and really, as we mentioned, this is really to help find and evaluate key research resources. And there are some questions that someone might need to answer. So, for example, if you're publishing a paper, you might be requested for research resource identifier, to cite key resources. And this section of the portal is what would assist a researcher in doing that. So the research reports are basically built around information from a number of community authorities on these resources. So for antibodies, there's an antibody registry where vendors, labs contribute information about their antibodies. For example, for plasmids, we get this information from Adgene, which is a large plasmid supplier. For model organisms, there are a number of different databases out there for model organisms that are maintained by the community, such as Jackson Labs, the Rat Genome Database, Flybase. These are all community-run authorities for specific organisms, and that information is brought into these reports, for example, surrounding organisms. And the reports include basic information about and a resource. So in this example, you're seeing information about an antibody, but we also are able to pull out who is using them. A lot of times in some of these resources, there's also information about potential issues. For example, for antibodies, has an antibody been discontinued? Is it non-specific? And also, how do they compare? So we can basically, use these reports to, assist in, one, deciding, is this the appropriate antibody, that I should try? Are there others that people are using? So for example, within the literature itself, as we track how antibodies are used, we can also see, what other antibodies are used, with this antibody. And also, since we're getting information from PubMed, we can also get information about location. So is anyone near me familiar, with this antibody? And this is one of the sort of the newest features for these resource reports that we're starting to test and roll out. And the back of all this information about resources is that, by pulling in this information from, the resource authorities that exist out there, pulling information from the literature, pulling information from other large projects, for example, that do validatings and ratings, such as the Human Protein Atlas and ENCODE, which provide additional information about antibodies. We're able to, assemble this unique data set, which aggregates all this information about these key resources. And what ties this all together is the research resource identifiers. So I'm not sure if you've heard about research resource identifiers, but they are persistent identifiers for research resources. And so a number of years ago, there were, and there still are, issues in terms of identifying, antibodies, model organisms, in the literature, in that the original way that journals required to report this information, for the majority of the citations for these resources, one could not go back, to the vendor and find the exact same antibody or organism that was used, just in terms of the way that it was written in the paper at that time. So this idea of having a unique identifier for these resources, was pitched to the journals, and it has now gained a widespread adoption. We were one of the groups that was instrumental in development and adoption of these RRIDs, and it really is, for the author then to supply, and identify the exact resource, in the materials and methods section, in terms of what they used. And this is now also incorporated, for example, in Cell Press's STAR methods, which are enhanced methods reporting requirements, for identification, of what was used in the paper. And again, the main reason that this was done was, to answer what resources were used in the study, but also who else is published, with this resource, and makes then, tracking, of, the antibody, seeing who else has used it, much easier. So the RIDs themselves are now, supported across major publishers, including Cell Press, Elsevier, Wiley, and Nature. They're in more than 1,000 journals. And most recently, they were made part of the JATS standard, which is the standard interchange format for journals to supply information to PubMed. And more than 200,000 resources have been identified, in the papers that have the RIDs. And as I mentioned before, one of the key things in terms of having an RID to actually specifically identify a resource is that we're able to pull resource ratings and alerts, and we're able to aggregate that information. So, for example, for cell lines, we can see if for example, the International Cell Line Authentication Committee, where we're pulling data from, is registering that this is a problematic cell line. If there's validation information, for example, at the Human Protein Atlas, we can point people to that. If the antibody registry is saying that the antibody is discontinued, we can give researchers that alert. So again, provides, we try to provide not just information about the basic aspects of these resources, but also potential issues, things that researchers need to be aware of when they're planning their study or using these. And one of the important aspects of that is that this information network around resources, when one inserts it into the publication process, it actually allows people then to get this information, as they're, publishing their studies. So, the RRIDs, are provided through a single portal, which we maintain. And this provides then a way, to bring some of this information about these resources sort of into the process when people are working on publishing their results. And what we've seen is that the impact of RIDs, in one paper, we looked at use of problematic cell lines. This has always been, it's a very known aspect of the use of cell lines, that a lot of the cell lines being used, have issues, either in terms of the cell line, drifting, or there's a mischaracterization of the cell line. And when we looked at papers where RIDs were associated with cell lines, we saw a dramatic decrease, in the use of problematic cell lines. Now, the question, that doesn't say that, the RIDs, have, cause those cell lines not to be used, but potentially, what this does do is, authors become more aware, of, issues with the cell lines. that they are then, they have to potentially, redo some of the experiment, to use cell lines that don't have issues, or they need to, describe why, the cell lines, are not as affected. And so, the earlier we move, the use of, informational tools that can provide, this type of information to researchers, the better it will be. And that sort of leads us into, the next section, which is the authentication reports that we're providing. So here, when you're planning your experiment, and you want to learn how to identify and validate resources, we can use that same information and alert researchers of any issues, with cell lines, and antibodies before the study is actually performed. So getting earlier in the experimental process, and this ties into NIH's Rigor reproducibility guidelines, where authentication is a key part of this, and the need, for researchers to develop authentication plans that they must submit with their proposals. In addition to, at the end of a study, to make your data shareable. So, this is a large area for NIH. In terms of the validation and the authentication plans, we provide authentication reports for cell lines and antibodies that provide information. For example, here what you see is that, there have been a number of, on the left, you'll see cell lines. On the top, you'll see antibody that has been selected. And, this has, again, in this report that's being prepared for the researcher in terms of, these are things that I've planned to use, there is information about an issue, with that antibody or cell line. And in addition to that, in providing that information in the report, we also provide guidance about validation. So there are a number of groups that are providing, best practice information. So what we do is we have aggregated that information, made that available, in these reports. And then, as you can see on the left, down the bottom, there's a list of references, for information about where we've gotten this validation information. So basically providing, guidance, assistance in terms of providing this information to NIH. The last bit of your experiment is then when, you're needed to share your data, make your data available. I mean, currently, the NIH guidelines, for data sharing are for projects that are greater than $500,000 in terms of direct costs, in terms of mandated sharing. But a lot of RFAs, are mandating data sharing by themselves, and there is a new data mandate that will be coming out of NIH, and more people, are going to be required to share their data. But the real issue, is, one, when you're writing your proposal, you need to know how to write, the data management plan. And this is an area where, actually libraries are already very engaged in terms of, providing assistance with data management plans. But then once, you're done with the research, where do you actually find a repository to share the data? So in terms of FAIR, this is a acronym that's been sort of picked up by NIH and NSF, and what it really means is, that data is findable, accessible, interoperable, and reusable, right? So that, there's sort of high-level aspiration as to what the data should be once it is made available to the broader community. And within dkNET, we're working to help interpret, these principles, these 4 principles, for the DK domains, and in addition to that, then providing tools to assist with that. As I mentioned, the fair data principles, are these, 4 high-level principles, to ensure that, once you publish a resource, other users can actually find, that the data is available, that, that users are able to access it in a usable form. And then if people want to take data from different repositories, that they can actually use that data with other data, do new analyses, do larger scale analyses. And this is not just for humans, but also machines. So for example, Google goes out and does searches for data sets across a number of different repositories, depending on what type of markup these repositories use. And so we also want, the machines to be able to also process this information, and to make it then easier to use. In terms of the FAIR attributes, so for each of these high-level principles, there is a number of different attributes for them, and many of them rely on metadata. So how rich is the metadata to describe a data set? What type of vocabularies are used, in terms of that metadata to make this data more interoperable? And then for reusability, that these metadata meet sort of domain relevant community standards. So there's a lot of emphasis on the metadata for data sets themselves, along with things such as globally unique and persistent identifiers, so that data can be found in the various repositories. And what we recognize is that there are a number of organizations that are involved in sort of this FAIR partnership, right? There are researchers, and a lot of times what they need to do for making their data fair is just starts with good data management and good documentation, right? So basically good lab practices, and then repositories, be they for example institutional repositories at a university, or a domain specific repository, for example one of the NIH NCBI data repositories that exist for genomic data. They're responsible then for issuing identifiers, making sure metadata is persistent, things of that nature. And then the indexers and aggregators, the Googles of the world, need to be able to provide effective search across those data sets. So in terms of what we're doing to assist researchers, the first thing is provide information about data management planning tools. As I mentioned, a number of libraries, groups are already providing assistance there. So what we're doing is making sure that we can link out to these various aspects in terms of preparing a proposal and providing information about the data management aspects of that. But once you've actually as I said, completed your experiment, where can one deposit data? And again, this is something that is a little more complex because, as I said, you have many different types of repositories that are out there. You have generalist repositories, such as Figshare and Dryad, where anyone can provide data. There are other repositories that are, supported or recommended by NIH institutes. And then there are also repositories that, are, listed, focused on specific scientific disciplines. Journals have other recommended lists. So it is a very sort of a complex world in terms of determining, where one might go. And so this is where, we're providing, based on, our discussions with NIDDK, where people should be depositing data, based on, a number of different criteria. This is one of the newer areas that we're building out as well. And so, over the next year, in addition to, some of these more basic tools, we're also going to be start implementing more of a walkthrough process where users can walk through, describe types of data they have, and we can actually then, suggest, what repositories they might want to go to. Oops, sorry, wrong way. As part of our outreach, so in terms of, many of the things that we've described, we're also providing as I said, information to students, about, the importance, of rigor reproducibility. And so we have a dkNET'S Summer of Data student internship program. Application deadlines are in just under 2 months. This is a summer project, program where we provide, students the opportunity, to use the tools and resources we have for their research projects. We provide information on best practices for rigor and reproducibility, and also, basics of good data management, following, the FAIR principles. And so really this is a program, to provide, training which they may not normally get in terms of, the specific focus on their research domain, but about, the important aspects of rigor, reproducibility, and good data management. As I mentioned at the very beginning, there are other aspects of the resource, in terms of providing ability to search information across, a large number of different biomedical data repositories. So in terms of doing some background research, and this is our discovery portal. And when one goes to the discovery portal, one can do a simple simple search, and then it returns information in terms of, what category of information is it? Is it literature? Is it data? Is it information on funding? Is it a physical resource? And provides then a list of, different data resources, information resources, that one can look at to, find out, if there's any information that one can use. One of the key things that we're doing is that, within the DK community, we've been aggregating information from, DK specific resources. So, information, for example, on, with HERN in terms of human islets, the mouse metabolic phenotyping centers, the beta cell consortium, and the like. And lastly, one of the newer areas of the portal is the Hypothesis Center. And really, this is, meant to provide a way for researchers to easily explore, for example, a lot of the omics data that is out there, but do it in a way, where a researcher can do that themselves and doesn't require, a lot of bioinformatics expertise to do this exploration. And so the Hypothesis Center, one of the initial tools that we've integrated with this is the Signaling Pathways project. And this is a knowledge base for cellular signaling pathways. And so, users can go in, and what the Signaling Pathways project does is they curate the data coming, for example, out of, one of the NIH genomics repositories, to make the data more fair, as we described earlier. And that allows them to basically provide a user interface where investigators, researcher students can, come in with genes of interest, look at receptor families, look at signaling pathways and how, genes in those pathways, are regulated. So again, this is one of the newer areas, but really, we're looking, to provide, tools, such as the Signaling Pathways project, to make it much easier for researchers to get in and explore some of this data to help them, generate new hypotheses. Covered some of the main areas of dkNET, and in terms of how sort of summarizing this, we had the resource reports, where researchers can use this to help find resources for their research, keeps researchers updated on resources. So if you actually go back, to your authentication reports, if information about an item has changed, for example, let's say a cell line, has been found to have issues after you've started using it, when you come back to your, your report, you'll actually see that it now has a warning. Also helps, for example, if you're using, a specific model organism, a specific antibody, you want some you know, information, from potentially from someone else who might have used that antibody. Again, we try to also help potentially find collaborators that might have used the same resources. The resource report information, again, gets put into these authentication reports, which helps with the authentication plans. And then, the fair data services are really, once the experiment, has been completed, how can I actually publish my data, in a data repository, the discovery portal connecting researchers to the broader biomedical community and the information resources that are available, and the hypothesis center, to enable researchers to really harness power of some of this big data that's out there without the need, for deep bioinformatics expertise. If one wants to explore, how, for example, the data relates genes to pathways and to other aspects. So I think that is, the end. We want really people to get involved in the dkNET community. So we do have, a webinar series that is running, where we provide information on various tools, various resources available to the community. And we record these webinars, and they're available on our YouTube channel. So that's another good resource. We have a newsletter, where we, monthly send out information about new resources that are available to researchers, new funding opportunities. So again, that's a good resource for investigators. And then, we have, our Twitter feed and other community news, where we provide information about other, community activities that may be happening. And at the end of this presentation, all the different areas that we introduced, there are links to all these different available resources that you can also use. So that's the end of my presentation. So just wanted to thank everyone. And if there are any questions, and then Koei can also go through some demos as well.
I have a couple quick. This is Stephanie. So will we be posting these slides somewhere so they can have access to these?
Yes, yeah. We can post those slides, and Koei can actually, can post those
And send out to everybody.
Yes.
And then I guess the only comment I would have, thanks Jeff, is we really encourage researchers and students to come here before they start their research projects, especially with the resource reports, because if they're going to have problems with an antibody or something that they're about to purchase, they can research it here first before they spend the money. So we're really looking at this as a place for researchers to come before they even begin their project. Even before, when they're submitting applications to the proposals, would be a good place to come. All right, so I'll turn it over to Koei, and if you have questions, you can put it in the chat or.
So I think I have to stop sharing, so I can turn that over to Koei.
Okay. Yeah, so if you have any question, you can put in the chat, or you can actually unmute yourself to ask question. So since we have some time, I'm going to give a demo of our portal. Let me share the slide first. So as Jeff just mentioned, there are different areas that dkNET portal can provide different service during their research process. At today's demo, I'm going to focus on plan experiment, which I'm gonna show a demo of resource report. And if we have more time, I'm gonna show you how to add a resource to the authentication report. So when a research comes to library, and if they are looking for information about resource report, or if they are going to publish a paper and they don't know how to cite a research resource correctly, you could direct them and show them they can go to the dkNET resource report to find the resource, to find the collaborator, and find how to cite this resource correctly. So to go to the portal, just type dkNET.org. you'll come to the portal, and there's 4 areas: resource report, discover portal, authentication report, and fair data, and hypothesis center. So when go to resource report, research can either directly collect resource report or go to a landing page, or they can just directly select the different research resource they want looking for. For example, if they are looking for cell line, they can click here. They are looking for an antibody, they can click antibody. So I'm going to go to the landing page first. So here are 6 different research resource type, including software tools, database, cell line, antibodies, organisms such as mouse, animals, different model organisms, and plasmid and biosamples. If research are looking for an antibody, they can go to antibodies. So they come to the search page. They can either type a keyword such as antigen, target antigen, or we suggest if they already know the vendor information and catalog number, they can input the information there, so it will be easier for them to find a specific antibody. So let's see if researchers are interested in finding an AKT antibody, which is an antibody that targets the AKT protein. They could do a search. Let me see. So here comes the result. As I mentioned, since there are thousands antibody that target AKT, so the result comes at 1,01457. So they could browse the result, switch between this snippet view or table view. Depend on the preference that they like. They could refine the result by using different facets. For example, if they already know what target organism they are interested in, they can click and by select, or they can select the clonality or host organism. And dkNET aggregate information such as who else have been using this antibody by mentions in open access. So actually it can help research to select the antibody. So we also have different facets about validation information and then issues that we have been found. So here, if I'm only interested in an antibody that have been mentioned in the papers, I'm gonna click filter out the antibody that only have mentions. So here shows you these resources have been mentioned more than 100 times in the open access papers. And the second antibody also mentioned more than 100 times. However, this one also have a rating and validation information available in certain community. So to reach the resource report, just click the antibody name. And here comes the resource report. So Jeff already mentioned in the presentation, there's different sections. I just want to show you that in the basic information, user can click more to see more information provided here. And then for the usage metrics, here shows there are 208 mentions in open access literature. User can click view full usage report to reach the usage report. At the usage report, here shows the mentions published by years, and then user also can find other frequent mentioned resource with this resource. There's different filter to allow them to filter all the mentions, and user can also download the mentions and to work offline. So back to the resource report, there's one section called Collaborator Network. User can find mentions based on their location. For example, if user type in California, by selecting that and click search. they can find a collaborator that in the California area. And then if they are looking for some experience from collaborators, they can contact them. Or if they need an antibody very urgent, so they can also contact to see if they have those antibodies available. Okay, let me go back to the resource report. And there's other section called rating and alert. So here shows this antibody have been rated by this consortium called Intestinal Stem Cell Consortium. User can click the information and we will bring them to the community resource and they can see the rating and the other validation information. And there is a small section called authentication plan. It's actually help users to go to authentication report section. So this is the resource report. Now I'm going to, since we have some more minutes, I'm going to show how research add their resource to the authentication report. So authentication report is a section that when a user comes to library and then they say, oh, I'm going to submit a grant to NIH system, and NIH requests some more information in supplemental, including authenticating the key biological resource. Then we provide them a report to help them to prepare that section. So user can click authentication report, and then there's some more information about NIH mandate if they are not familiar with, and how to create this report. So they can click the information. To create report, just directly click create a report. And here are basic information about how to prepare this authentication plan. And they can click start the report. So user need to have a dkNET account and it's free to create a report. That way they can save their resource in their account. So I'm going to create a report. By logging. So here is a dashboard about all my previous authentication report, but create a new report, they just need to click new authentication report and do the report name. Let me just do today's date. Submit. So here comes the report. It's the report name, I called it test 022620. And then they can start to select their resource that they would add to their report. For example, antibody, they can just type the same antibody that they are searching. And then if this is the antibody they are looking for, they can just add to the report. And here shows that AKT antibody is successfully added, so it will show under their report item. User can also add cell line, for example. If they are going to search for an HEK cell line. Okay. Here comes a list of cell line. And then you will see, as Jeff mentioned in the presentation, if this cell line have an issue, for example, HEK cell line, it's a problematic cell line. It has been showing in the cell source that it's contaminated. So our system will alert research before they even plan to add to their proposal or plan in their experiment. So if research didn't notice about this and they insist to add, the system also warned them it's successful added, but there is a critical issue. And if they are interested in what the issue is about, they can click the cell line on the report item and then found the information here. So let me report to the dashboard. Okay. So if research already add all their research resource items in their report, now they can preview their authentication report. So by clicking preview the authentication report, here shows you how this report look like. Research can use and copy the language for their proposal submission. We provide identification of cell line information and then how the authentication plan should be done in their lab. For antibody, we also provide different validation principles based on the paper that published by Woolen. So research can use this language and report while submit their proposal. So here are the two demos. I'm gonna stop my sharing, see if there's any question from you. So are there any questions? Or do you need any more specific area that I could show a demo?
Okay, thank you, Koei.
Mm-hmm.
So that sort of concludes our overview of what dkNET provides the community. Stephanie, did you have any final pieces of information?
No, I just want to thank both you and Koei for the presentation, and those on the call. Thank you so much for taking the time out to look at this. If you want any additional information or if I can help you at all, I think you've got my email, and we'll send you a little note as well. And then please feel free to, we'll have some more of these presentations in the future, and feel free to send it to your colleagues who may be interested in this resource. That's it. Thank you all. Oh, there's one chat. Oh, it looks great. Thank you, Diane. All right. Bye, everybody.
Bye.